A Malaysian-born couple and their 3-year-old son were found dead in their London flat on Tuesday, reported the BBC. Lead investigator Detective Chief Inspector Simon Harding said the case was being treated as a murder investigation.
Today, I and my partner are going to discuss about how the forensic experts find the murderer by using this technique called as DNA fingerprinting. DNA fingerprinting is known as a method that is used to identify an individual from a sample of DNA by looking at a unique pattern in their DNA. This method was invented in 1984 by Professor Sir Alec-Jeffreys after he realized that the variation in human DNA can be detected in the form of mini satellites. The probability of having two people with the same DNA fingerprint that is not identical twin is very small. Just like your actual fingerprint, your DNA fingerprint is something that is born with you. It is unique to you.
I'll be giving further explanation about how a DNA fingerprint produces. So the first step, we need to extract a DNA from a human sample. Human sample could be blood, saliva, nails, or even a hair strand. Then a molecular scissors called restriction enzyme were used to cut the DNA. This produces thousands of DNA with variety of length. Then these pieces of DNA were separated according to their sizes by a process called gel electrophoresis. Once the DNA had been sorted, the pieces of DNA were transferred out of the fragile gel onto a robust piece of nylon membrane and then unzipped to produce single strand of DNA. Next, the nylon membrane was incubated with radioactive probes. Probes. What are probes? Probes are small fragments of mini satellite DNA that packed with radioactive probes. The probes only attach to the pieces of DNA that are complementary. The next step is the mini satellites that the probes attached were then visualized by exposing the nylon membrane to an X-ray film. When exposed to radioactivity, a pattern with 30 dark bands appeared on the film. This pattern called DNA fingerprint. To compare 2 or more DNA fingerprints, the different DNA samples were run side by side on the same electrophoresis gel. For the next part, we will be talking about modern-day DNA profiling. Modern-day DNA profiling is also called as STR analysis and relies on microsatellites rather than mini satellites that used in DNA fingerprinting. Microsatellites or short tandem repeats STR are the shorter relative of mini satellites, which is usually 2 to 5 base pair long. Like mini satellites, they are repeated many times throughout the human genome. For example, TA, 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 TA. Now, my partner will be explaining about how DNA profiling produced today.
How is DNA profile produced today? DNA is extracted from a biological sample. The STR analysis is incredibly sensitive, so it needs only a tiny amount of DNA to get an accurate result. Unlike the original DNA fingerprinting, DNA profiling does not use a restriction enzyme to cut the DNA. Instead, it uses a PCR, which is a polymerase chain reaction, to produce a many copy of specific STR sequences. As a result, DNA can be extracted from a biological sample, including hair, saliva, and blood. PCR is an automated procedure that generates lots of copies of a specific sequence of DNA. It only requires small amounts of DNA to start with and can even make copies from a DNA sample that is partially degraded. In PCR. Small bits of DNA called primers bind to complementary sequences of the DNA of interest and mark the starting point for the copying of the DNA of interest. In STR analysis, the primers used in the PCR are designed to attach to either end of the STR sequence of interest. The primers for each STR is labeled with a specific color fluorescence tag. This makes it easier to identify and record the STR sequences of the PCR. Once enough copies of the sequence have been produced by PCR, electrophoresis is used to separate the fragments according to size. Each fragment passes by a laser which causes the fragments with fluorescent tags to glow with a specific color. The output is displayed as a series of color peaks, as shown in the image below, highlighting the color and length of each STR sequence. The more STR sequences that are tested, the more accurate the test is at identifying someone. Other STRs used for forensic purposes are called Y-STRs, which are derived solely from the male Y chromosome. This is useful for identifying a male perpetrator from mixed DNA samples. Only one person in every 10 million will have a particular STR profile, with the world human population estimated at only 700 million. It is therefore extremely unlikely you will share the same profile as someone else unless you are an identical twin.